Welcome back to Concepts of Biology.
We'll read section 4.3, Citric Acid Cycle and Oxidative Phosphorylation. By the end of this video, you will be able to describe the location of the citric acid cycle and oxidative phosphorylation in the cell. Describe the overall outcome of the citric acid cycle and oxidative phosphorylation in terms of the products of each, and describe the relationships of glycolysis, the citric acid cycle, and oxidative phosphorylation in terms of their inputs and outputs. The citric acid cycle. In eukaryotic cells, the pyruvate molecules produced at the end of glycolysis are transported into mitochondria, which are the sites of cellular respiration. If oxygen is available, aerobic respiration will go forward. In mitochondria, pyruvate will be transformed into a 2-carbon acetyl group by removing a molecule of carbon dioxide that will be picked up by a carrier compound called coenzyme A, which is made from vitamin B5. The resulting compound is called Acetyl-CoA. Acetyl-CoA can be used in a variety of ways by the cell, but its major function is to deliver the acetyl group derived from pyruvate to the next pathway in glucose catabolism. Figure 4.14. Pyruvate is converted into acetyl-CoA before entering the citric acid cycle. Like the conversion of pyruvate to acetyl-CoA, the citric acid cycle in eukaryotic cells takes place in the matrix of the mitochondria. Unlike glycolysis, the citric acid cycle is a closed loop. The last part of the pathway regenerates the compound used in the first step. The 8 steps of the cycle are a series of chemical reactions that produces 2 carbon dioxide molecules, 1 ATP molecule or an equivalent, and reduced forms NADH and FADH2 of NAD positive and FAD positive, important coenzymes of the cell. Part of this is considered an aerobic pathway or oxygen requiring because the NADH and FADH2 produced must transfer their electrons to the next pathway in the system, which will use oxygen. If oxygen is not present, this transfer does not occur. 2 carbon atoms come into the citric acid cycle from each acetyl group. 2 carbon dioxide molecules are released on each turn of the cycle. However, these do not contain the same carbon atoms contributed to the acetyl group on that turn of the pathway. The 2 acetyl carbon atoms will eventually be released on later terms of the cycle. In this way, all 6 carbon atoms from the original glucose molecule will be eventually released as carbon dioxide. It takes 2 turns of the cycle to process the equivalent of 1 glucose molecule. Each turn of the cycle forms 3 high-energy NADH molecules and 1 high-energy FADH2 molecule. These high-energy carriers will connect with the last portion of aerobic respiration to produce ATP molecules. 1 ATP, or an equivalent, is also made in each cycle. Several of the intermediate compounds in the citric acid cycle can be used in synthesizing non-essential amino acids. Therefore, the cycle is both anabolic and catabolic. Hi! If you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. All right. Let's get back to reading. Oxidative phosphorylation. You have just read about 2 pathways in glucose catabolism, glycolysis and the citric acid cycle that generate ATP. Most of the ATP generated during the aerobic catabolism of glucose, however, is not generated directly from these pathways. Rather, It derives from a process that begins with passing electrons through a series of chemical reactions to a final electron acceptor, oxygen. These reactions take place in specialized protein complexes located in the inner membrane of the mitochondria of eukaryotic organisms and on the inner part of the cell membrane of prokaryotic organisms. The energy of the electrons is harvested and used to generate an electrochemical gradient across the inner mitochondrial membrane. The potential energy of this gradient is used to generate ATP. The entirety of this process is called oxidative phosphorylation. The electron transport chain is the last component of aerobic respiration and is the only part of metabolism that uses atmospheric oxygen. Oxygen continuously diffuses into plants for this purpose. In animals, oxygen enters the body through the respiratory system. Electron transport is a series of chemical reactions that resembles a bucket brigade in that electrons are passed rapidly from one component to the next to the endpoint of the chain where oxygen is the final electron acceptor and water is produced. There are 4 complexes composed of proteins labeled 1 through 4 in figure 4.15c, and the aggregation of these 4 complexes together with associated mobile accessory electron carriers is called the electron transport chain. The electron transport chain is present in multiple copies of the inner mitochondrial membrane of eukaryotes and in the plasma membrane of prokaryotes. In each transfer of an electron through the electron transport chain, the electron loses energy, but with some transfers, the energy is stored as potential energy by using it to pump hydrogen ions across the inner mitochondrial membrane into the intermembrane space, creating an electrochemical gradient. Figure 4.15. A. The electron transport chain is a set of molecules that supports the series of oxidation and reduction reactions. B. ATP synthase is a complex molecular machine that uses a hydrogen gradient to regenerate ATP from ADP. C. Chemiosmosis relies on the potential energy provided by the hydrogen gradient across the membrane. Here's a situation. Quick question. Put your answers in the comment section below about this. Cyanide inhibits cytochrome C oxidase, a component of the electron transport chain. If cyanide poisoning occurs, would you expect the pH of the intermembrane space to increase or decrease? What effect would cyanide have on ATP synthesis? Put your answers in the comments below. Let's have a conversation. Electrons from NADH and FADH2 are passed to protein complexes in the electron transport chain. As they are passed from one complex to another, there are a total of 4. The electrons lose energy, and some of that energy is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. In the 4th protein complex, the electrons are accepted by oxygen, the terminal acceptor. The oxygen with its extra electrons then combines with 2 hydrogen ions, further enhancing the electrochemical gradient to form water. If there were no oxygen present in the mitochondrion, the electrons could not be removed from the system and the entire electron transport chain would back up and stop. The mitochondria would be unable to generate new ATP in this way, and the cell would ultimately die from lack of energy. This is the reason we must breathe to draw in new oxygen. In the electron transport chain, the free energy from the series of reactions just described is used to pump hydrogen ions across the membrane. The uneven distribution of hydrogen ions across the membrane establishes an electrochemical gradient owing to the hydrogen ions' positive charge and their higher concentration on one side of the membrane. Hydrogen ions diffuse through the inner membrane through an integral membrane protein called ATP synthase. This complex protein acts as a tiny generator turned by the force of the hydrogen ions diffusing through it down their electrochemical gradient from the intermembrane space, where there are many mutually repelling hydrogen ions, to the matrix where there are few. The turning of the parts of this molecular machine regenerate ATP from ADP. This flow of hydrogen ions across the membrane through ATP synthase is called chemiosmosis. Chemiosmosis is used to generate 90% of the ATP made during aerobic glucose catabolism. The result of the reactions is the production of ATP from the energy of the electrons removed from hydrogen atoms. These atoms are originally part of a glucose molecule. At the end of the electron transport system, the electrons are used to reduce an oxygen molecule to oxygen ions. The extra electrons on the oxygen ions attract hydrogen ions or protons from the surrounding medium, and water is formed. The electron transport chain and the production of ATP through chemiosis are collectively called oxidative phosphorylation. ATP yield. The number of ATP molecules generated from the catabolism of glucose varies. For example, the number of hydrogen ions that the electron transport chain complexes can pump through the membrane varies between species. Another source of variance stems from the shuttle of electrons across the mitochondrial membrane. The NADH generated from glycolysis cannot easily enter mitochondria. Thus, electrons are picked up on the inside of the mitochondria by either NAD positive or FAD positive. Fewer ATP molecules are generated when FAD positive acts as a carrier. NAD positive is used as the electron transporter in the liver and FAD positive in the brain, so ATP yield depends on the tissue being considered. Another factor that affects the yield of ATP molecules generated from glucose is that intermediate compounds in these pathways are used for other purposes. Glucose catabolism connects with the pathways that build or break down all other biochemical compounds in cells, and the result is somewhat messier than the ideal situations described thus far. For example, sugars other than glucose are fed into the glycolytic pathway for energy extraction. Other molecules that would otherwise be used to harvest energy in glycolysis or the citric acid cycle may be removed to form nucleic acids, amino acids, lipids, or other compounds. Overall, in living systems, these pathways of glucose catabolism extract about 34% of the energy contained in glucose.
Let's look at a career in action.
Mitochondrial disease physician. What happens when the critical reactions of cellular respiration do not proceed correctly? Mitochondrial diseases are genetic disorders of metabolism. Mitochondrial disorders can arise from mutations in nuclear or mitochondrial DNA, and they result in the production of less energy than is normal in body cells. Symptoms of mitochondrial diseases can include muscle weakness, lack of coordination, stroke-like episodes, and loss of vision and hearing. Most affected people are diagnosed in childhood, although there are some adult-onset diseases. Identifying and treating mitochondrial disorders is a specialized medical field. The educational preparation for this profession requires a college education, followed by medical school with a specialization in medical genetics. Medical geneticists can be board certified by the American Board of Medical Genetics and go on to become associated with professional organizations devoted to the study of mitochondrial disease, such as the Mitochondrial Medicine Society and the Society for Inherited Metabolic Disease.
This ends Chapter 4, Section
4.3, Citric Acid Cycle and Oxidative Phosphorylation.
In the next video, we'll read Section
4.4, fermentation.